Hi everyone, welcome back to the Biotechnology series. We are here. Yes, this is the final lecture of the Biotechnology series. I guess we could call it the Season 1 finale. Now this lecture is not very typical because I am going to talk about the ligand evolution research that I have been doing with my students and myself when I was a graduate student, and how it originated from people studying the origin of life. I believe this is a unique lecture that you won't hear anywhere else, so let's get started. So because of this special nature of this lecture, there aren't any real objectives. I just hope if you're listening, pay attention, stay awake, and watch the entire video. How about that? So in this two-part series, our last episode, we talk about how directed evolution helps us to generate new enzymes and also screen for new antibodies or antibody fragments. Now, in this episode, we are going to talk about different kinds of biomolecules that are capable of binding and how they are going to be discovered, and what are their properties, and what are their applications, and can we even take them to the next level. So it is all about past, present, and future. Now let's have a little history lesson and look at the past. That means the pioneer of the field. Now, generally, we've been talking about all these binding molecules, no matter it is the antibodies or antibody fragments. Well, they recognize some target, right? So we give them a broader term. We call them molecular recognition elements, or MRE. So they can be small peptides through phage display. They can be antibodies. Now, they can also be single-stranded DNA and RNA, and I'll explain why. Now, these molecular recognition elements can all bind to a target. Now, these targets can be proteins, small molecules, viruses, or even whole bacteria or mammalian cells. And when this binding occurs, we always want them to have high affinity and specificity so that they could be useful. So when these molecular recognition elements are made of nucleic acids, well, we have a special name for them. They are also known as aptamer. Now this aptamer term is a put together word with a Latin word, aptus, means fit, and a Greek word means part, mers, okay, aptamers, but that means a fitting part. Now, they can be oligonucleotides, and sometimes small peptides can also be categorized as aptamers. And by definition, they can bind to a specific target molecule with high affinity and specificity. Now, how do you get them? It is through a process called in vitro selection process. Now, this process is very much like what we've just talked about in the part 1 of this lecture. Now, this whole process, or this term, actually was first described by two scientists, Dr. Andrew Ellington and Dr. Jack Szostak. Now, Dr. Jack Szostak, actually, he was the 2009 Nobel Prize laureate in physiology or medicine because of his work in the origins of life. Now, their work in aptamer actually was published in 1990 in Nature, and I have their paper referenced there. So how does in vitro selection of an RNA molecule related to the origins of life? Now, the question of the origin of life and early evolution of life has fascinated scientists for centuries. One prevailing theory, known as the RNA world hypothesis, suggests that the first biological catalyst arose from a random sequence polymers, such as polynucleotides and peptide chains. However, the chances for these random sequences forming stable 3D structures with binding or catalytic activity are believed to be very low. So in order to study the evolutionary process of RNA molecules, scientists developed an in vitro method to quickly speed up or direct the pool of random RNA for a specific target. And through this in vitro selection process, they isolated RNA sequences from random pools of up to 10 to 13 different molecules with high ligand binding affinity, suggesting the possibility of isolating novel ribozymes or catalytic RNA from a random pool. Now, this discovery has added weight to the RNA world hypothesis, which holds that in the primordial soup, free-floating nucleotides responsible for the earliest biological catalytic functions on Earth, or maybe elsewhere. Now, despite ongoing debate and research, the question of the origin of life and early evolutions of life remains a topic of great mystery and scientific inquiry. Great science always happened at about the same time. Now remember, Dr. Ellington and Dr. Sostak published their work in the journal Nature in 1990, and right about the same time, two other scientists published a very similar work in the journal Science, also in 1990. Now they are Dr. Turk and Dr. Larry Gold. They're another group of scientists who conducted a very similar experiment to isolate RNA ligands. And in their research, they selected RNA ligands to bacteriophage T4 DNA polymerase and termed the in vitro selection process systematic evolution of ligand by exponential enrichment, or commonly known as SELEX. Now, this groundbreaking work led to the development of some commercial products because they were smart. They patented the commercializations of APTAMER and related process and products for 20 years. And they also founded a company called Next Star Pharmaceuticals in Boulder, Colorado, which focused on the development of APTAMER-based therapeutics. And their work ultimately led to the approval of the first and only APTAMER drug, Macugen, for the treatment of age-related macular degeneration. Now today, their legacy continues through a new company known as SomaLogic, also located in Colorado, which specializes in development of technology using aptamer-based assays. Now let's look at the in vitro selection process for nucleic acids ligands. The systematic evolution of ligands by exponential enrichment (SELEX) process is a laboratory technique that allows the selection of RNA or DNA molecules with high binding affinity to a target molecule or biomolecules of interest. Now, the SELEX process typically involves several rounds of iterative selection and amplification, where a large pool of RNA or DNA molecules with random sequences is incubated with the target molecule. After incubation, any molecules that have bound to the target are separated from those that have not, and the bound molecules are eluted and amplified using polymerase chain reaction or reverse transcription. Now, a similar process can add into the whole SELEX process to incubate with a so-called negative target or target that we do not want to bind. Now, during which those that bond to the negative target are removed, and those that do not bind to the negative targets or target that we don't want to bind are retained and amplified similarly. And the amplified pool is then subjugated to other rounds of selections, so it basically repeats until the enriched pool contains a high percentage of molecules that bind specifically to the target. And finally, the selected RNA or DNA molecules are sequenced to identify the sequences of the specific molecules that have high binding affinity to the target. Now, this process I've done many times during my graduate school, and I've also taught several of my graduate students do this process. So notice that two separate groups of scientists were developing the same process and generating similar molecules. So clearly there was a rivalry between Dr. Andrew Ellington and Dr. Larry Gold. And the rivalry between them were over the development and commercializations of aptamers. And in 1990s, Dr. Gold's group at Nestor Pharmaceuticals developed the first aptamer drug, Macogen, like we said before, which was approved by the FDA. Meanwhile, Dr. Ellington's group at the University of Texas at Austin was also working on aptamers and developed a different selection method. Well, now that led to some legal dispute. Now, it was smart that, for Dr. Gold, who patented things first, so he got the first dibs, even though Dr. Andrew Ellington were a little bit ahead in terms of generating the data. So, basically the legal battle were between the two groups over, commercializations and potential, commercial interest. So it was quite, intense in a sense of speaking. Now, the dispute eventually led to a settlement in which the right to use for aptamer for certain diagnostic applications were granted to Dr. Ellington's group, and the right for use to as aptamer for therapeutic applications were granted to Nestor Pharmaceuticals. Now, however, the rivalries between them continued, which seek to develop and patent new aptamer-based technology. Now, since Dr. Andrew Ellington's group were working more so on the diagnostic side of the story, which really led to what I've been doing. Now, so anecdotally, there was a phone call conversation between the two scientists, and someone told me that actually Dr. Ellington were kind of yelling on the phone at Dr. Larry Gold. It was quite intense. I wouldn't describe the word, but it was mad word. Now, how I get to know their conversations? Well, I wasn't there, but someone I know was there, and I'll tell you why. Just follow along with the lecture. So now we knew about the past of this ligand evolutions of nucleic acids. Now let's look at the present, or a little bit more present from my perspective. So this is purely my perspective. If you listen to another aptamer scientist, he or she may have different perspective. So this is a very personalized lecture in my opinion. So we've been talking about a selection begin with a large random pool of molecule. So what is this random pool? Now we call them a library. The overall library lengths are usually designed to be about less than 150 bases in total length, 150 bases of either RNA or DNA. And it has a random regions, usually between about 20 to 80 bases, and they are chemically synthesized using standard phosphoramidite chemistry. Now, anyway, so the random region gives rise to the diversity of the library, which can be designated as 4 to the nth, where nth is the number of bases in the random region. Now, so the longer the random regions results in increased library length and diversity, but the longer the nucleotide or oligonucleotide, the more difficult to synthesize. And when the overall length is over 80 bases long, the yield for full-length oligonucleotide is very low during the synthesis process. So in which usually, our library is around 80. Now, my library, yes, I do have a library to do this type of experiment, is actually 80 bases long. So now inside this design, along with the random region, there's also two constant regions, also called the primer annealing regions. So with the known regions, we can use polymerase chain reactions to amplify this whole molecule with traditional method. So here are a couple of figures in this slide, and those actually looks like a straight line on paper, but in reality, all these nucleic acids, even though they are single-strand, they can still fold in a three-dimensional shape, and this three-dimensional shape can convert to a binding pocket. And on the right-hand side, it shows a crystallized interaction structure between an aptamer that binds to its target, which is thrombin protein. So over the past 30 years, a lot of different aptamers have been selected or identified or screened, and they have been investigated in various applications, not just limited to diagnostics and therapeutics. Now, the only difference is that, well, researchers can do all they want with research. They just cannot sell them. Okay. Now, these applications include research applications, biosensors, drug discovery, biomarker discovery, and also in synthetic biology. So we're going to pick a few to talk about here. So first is therapeutics. Okay. Now, therapeutics early publications in this area, there's a very nice review, okay, by Dr. Allington's group. Even though he could not sell those things, him and his student at the time, Dr. Letha Suter, wrote a review article and published in 2002, which is, well, 21 years ago now. This is 2023. So a little reflections on novel therapeutic candidates. And I also wrote a mini review on the potential of using aptamers for thrombotic events treatment and monitoring back a few years ago in 2019 with one of my students. The therapeutic usage of aptamers in treating thrombotic events or even monitoring is an emerging area, and many different RNA and DNA aptamers have been selected targeting various factors in the colon cascade, including the VWF, factor 2A, which is thrombin, and also other factors, factor 9A, 10A, 11, 12A, and all those. And of all of those candidate aptamers, one of the most successful one was called REG1, which is an RNA aptamer targeting factor 9A. It went all the way to phase 3 clinical trial, but unfortunately it failed. Now, what was the reason? It was due to anti-PICOLINIC antibody. So PICOLINIC, we talked about somewhere probably, and PICOLINIC of a biomolecule actually is to increase its half-life so that it can stay in the body longer and do its work. Unfortunately, the body can also generate antibodies to target these PICOLINIC molecules. With that, it will render the drug molecule useless. So for those of you who would like to read on this topic, this is the table from my publications showing you different type of DNA and RNA aptamers that were in clinical or preclinical trials trying to target different targets in the clotting cascade. Besides treating thrombotic events, aptamers were also investigated extensively in using to treat cancer and also infectious diseases. Now, why is that? Because both cancers and infectious disease, there is a clear target that is different than our normal cells. Cancer cells are those that are abnormal and express cancer antigens, which are the ideal candidates. Now, if you want to learn more about cancer and immunology, I have another video there. Well, if you forgot about it, well, check that out. Anyway, infectious disease also have a clear target. You have a virus, you have a bacteria, and they are distinctively different than everything inside our body. Here we're just looking at an example of an idea of how to use aptamer as a nanodrug to treat cancer. Now, basically, the idea is very similar to antibody-drug conjugates, so you can check that lecture out again. Now, the idea is to link the aptamer with a therapeutic drug, and through this process, the aptamer will basically target the cancer-exclusive antigens and be internalized. Now, sometimes you can also add on different molecules to this whole assembly, such as adding gold nanoparticles in this whole assembly, and with something called near infrared frequency, you can actually heat up the gold nanoparticle that is being uptaken by the cancer cells. And with this heat up process, it can actually, burn the cancer cells. So a lot of these things are in investigation. So here just an example of that. Another way for aptamer as an therapeutic is as a neutralizing agent, very much like an neutralizing antibodies. Now we know that antibodies can neutralize bacterial toxins. Well, what about aptamers? Now in fact, it is possible. Now, bacterial toxins, again, they are virulence factors and they play a significant role in pathogenesis. And there are many aptamers that can competitively bind to the bacterial toxins. And here is a graph of a published model that is in the past showing you, well, this is a Staphylococcus aureus alpha toxins. And if you treat or neutralize the toxins, you can actually speed up the wound healing process. So what if you have an anti-alpha toxin aptamer that may have the usage, may also have the effect to speed up the skin healing process. However, we don't know if that would actually happen. But on the other hand, during my graduate work, I actually published an aptamer that can target alpha toxins from Staphylococcus aureus. So during some point of this biotechnology series, I've probably mentioned that my research focus, my work has always been in using biotechnology for diagnostic. Well, so precisely I'm using aptamers for diagnostics. So when I graduated, I published a very extensive review article with my advisor, Dr. Letha Suter, titled Single-Stranded DNA Aptamers Against Pathogens, Toxins, Identifications, and Biosensing Applications. So it was a very extensive amount of research went into this work. And later on in 2020, I also updated this work with my former student, Dr. Chunzo there. Now, anyway, so if you notice the name there, well, there's some connections, right? Dr. Letha Suter was Dr. Ellington's student, and I was Dr. Letha Suter's student. So there is that connection. So in terms of diagnostic applications, early works were mostly on protein targets, but there is a huge increase in selections of aptamers against bacterial host cells since 2007. Now, a lot of these bacteria are contributors to food poisoning, and it is important to have a very sensitive way to detect these organisms in food samples. Now, besides host cells, common non-specific bacterial cell surface molecules, such as these peptidoglycan or lipopolysaccharides, were also targeted for selections. So that is on the bacteria side. Now, for diagnostics and viral diagnostic in particular, HIV viral proteins were selected, but more so for therapeutics. But on the other hand, for influenza A viruses, those were all investigated in both therapeutics and detection applications. So what I'm showing you here is a gram-negative stain of Pseudomonas aeruginosa. So this is actually my photograph. I stained this when I was doing my PhD. And besides detecting the whole organisms, toxins, particular bacterial toxins and other fungal toxins, are also common target. Now, for bacterial toxins, major contributors, such as cholera toxins and cyanobacteria toxins, were frequently selected for its aptamer. The idea is for rapid detections. And for fungal toxins, there's a huge interest in trying to detect ochratoxin A or OTA. Now, there were over 50, probably 60, 70 journal articles published on OTA detections alone using aptamers. And also another toxin that is related is alpha toxins B1. Now, these toxins were commonly found in contaminated peanuts. So having a rapid way to detect them is crucial and very useful. Now, other than detecting bacterial toxins in food samples, we can also try to detect toxins in clinical samples using aptamer. Now, here is an example. Toxin B is a virulence factor of Clostridium difficile and account for many cases of diarrhea in hospital settings, and it is costly and also deadly. Clostridium difficile is actually a natural inhabitant of the intestine. However, when you have an overgrowth of the toxin-producing string, it will cause problem, and therefore detecting the toxin is relevant. Now, this toxins can inhibit members of the Rho family GDPase. Basically, it's just going to cause intestinal inflammations and excessive and severe watery diarrhea. Now, current diagnosis for the conditions, we have enzyme immunoassays, PCR cell culture, and confirmed diagnosis require multiple tests. So, having more options to diagnose or detecting this toxin may be useful for aiding rapid diagnosis. So, the idea is, can we find a aptamer for this toxin? And surely, yes. If I say that, that would be yes. That was also part of my graduate work. This was published in 2015. This is a specific aptamer that can target toxin B. So remember, I said toxin B can be found in fecal matter. Well, diarrhea, fecal matter. So when I was doing this work, I actually had to use real human fecal samples to test the methodology to develop this detection assays. Well, it wasn't exactly fun, but I did document the whole process with this. So sacrifice for science. So here is a brief schematic of the detection assays using aptamer as a capturing molecule and mimic a ELISA-like assays. Now, we talked about ELISA in earlier introduction lectures in the biotechnology series, so if you forgot about that, you can check that out again. Anyhow, so instead of having an antibody to capture the toxins, I have an aptamer to capture the toxins and use a sandwich fashion to sandwich it out and detect the toxins. Well, here is just the schematic. I'm not going to go with too much of a detail, but basically the detection assays were okay, quite relevant. It can detect a pretty low amount of toxin B and was sort of clinically relevant. Now, besides using aptamers to detect toxins or other infectious disease markers, we can also use aptamers to monitor drug concentrations in plasma, potentially, such as antibiotics, anticoagulants, or other drugs that have narrow therapeutic windows. Now, the idea is to develop a bedside diagnostic device such as lateral flow assays to closely monitor these drugs. So something in the work, that is currently in the work that I'm still working on the project to identify a single-stranded DNA specific to a direct factor 10A inhibitor. So I'm not going to say which drug yet, so hold that thought there. So while we are on the topics of lateral flow assays and other detection platforms, the bigger schemes of sensing or diagnostics, actually biosensing of not just detecting clinically relevant molecules, but also trying to detect other things that are in the environment. So several years ago, many years ago now, I was invited to write a chapter for a book entitled An Overview of DNA-RNA-based Monitoring Tools and Biosensors and Benefits and Applications in the Environmental Toxicology. That's how I got involved with all these pesticides and herbicides in my work. So I'm not exactly an engineer, but my work is always involved in identifying the binding molecules. But in more recent years, actual engineer or electrical engineer or bioengineers have been researching using single-stranded DNA aptamers in biosensors, and they basically developed various ways to detect these environmentally relevant molecules. And these detection methods can be categorized with, basically electrical, electrochemical, or other optical means using fluorescent absorbance or gold nanoparticles. Now, more recently, I've been more involved in using gold nanoparticle to detect my analyte. So I'm moving to that direction. So here are some of the examples of these environmentally relevant chemical toxins, such as 17-beta estradiols. Now, some of these can be from pharmaceuticals that we excrete and got into the water bodies. Second one would be bisphenol A, BPA. Now, this is a chemical in plastic productions. It can actually mimic estrogen effect, so it's quite important. Another one is polychlorinated biphenyl, which is PCB, used in various industrial settings and shown to contaminate water bodies and food sources. And other things such as herbicides and pesticides are always something of interest, such as atrazine, malathion, bromacil, fipronil, and many, many more. So I had done some work related to atrazine detections. Now, atrazine was outlawed in the Europe, but we are still using it in the US, although it's phasing out. Now, it's used in crop productions, mainly for corn. It's used to prevent growth of broadleaf weeds, and it can also be used on turf and lawns. Now, the problem with that is that atrazine persists in the environment and has shown to contaminate both groundwater, surface, and drinking water. And if you have a filter for your refrigerator, you would have noticed that those refrigerator filters claim to be able to remove some amount of atrazines. Now, atrazine is also a known hormone disruptor and has been linked to birth defects and cancers. So I mentioned moving into using gold nanoparticle for detections of toxins, and here are just some of the examples that I published a little more recently, 2018, 2019, showing how to use APTAMERS against atrazine incorporated with gold nanoparticle and detect atrazines in water samples. So basically the purple color is showing the detection, and also show a gradient change of detections from more pink to pretty dark purple. And finally, I'm also working on something else, another herbicide currently that is in the category of picolinic acid herbicide. Now, the problem with this type of herbicide, not so much with human health or animal health, but rather it is resistant for hydrolysis and photolysis, so they persist in the environment, even has been shown to contaminate compost pile. Now, if a compost pile have these herbicides, if you dump it back into the field as a fertilizers, it's going to damage crop. So having rapid ways to detect these herbicides using APTAMER potentially is very helpful, rather than sending the whole sample to the lab using LCMS to detect it. So that is something currently in the work. I'm not going to tell you which herbicide just yet, and wait until I publish the work. And to wrap up this lecture, well, a couple slides to wrap up, is that I have to acknowledge everyone that has contributed and has made all my work possible. And first, I need to acknowledge my mentor and my mentors, mentors, and mentors. And here is the academic pedigree. Now, so I have some remote connections with Dr. Szostak. So Dr. Andrew Ellington was Dr. Szostak's postdoc, and my mentor, Dr. Lee Thesuter, was Dr. Andrew Ellington's student at UT Austin, and I was fortunate to be Dr. Lee Thesuter's student at West Virginia University, and I graduated from her lab in 2015 and got my PhD. So it's some years now, thinking back, 2015, right? So I am sort of close to Nobel Prize, but not there. Certainly not there. Here is my graduating photo with my advisor, May 2015 in Morgantown, West Virginia. Now, here was my acknowledgement. Now, my thesis, my dissertation is published, and you can find out whatever I wrote there is no secret. But I'm also telling you one of my secrets here, that I am a big fan of Star Trek. Well, I don't know your age anymore. But anyhow, if you are a Star Trek fan, leave a comment. Let me know. Recently, the show just wrapped up with Star Trek Picard, and I'm a big The Next Generation fan, and I was so excited to see they recreate the Enterprise D bridge, the whole set. Well, just so good. Anyhow, so I had it a sentence in my acknowledgement in my dissertation that make the the motto in Star Trek. They said science the final frontier, right? We are the crew of the Enterprise and we explore space. And here I had it, science the final frontier. We are the members of the laboratory of Zudo, and our continued mission is to explore strange molecular worlds to boldly select binding elements which no one has selected before. So sort of like to boldly go where no one has gone before. And here is more acknowledgement. So a lot of my work was done when I was at Wilkes University, still with a lot of my former pharmacy students, and many of them have all graduated, and I also have some students that are still in school doing trying to get their PharmD. All right, here on the list. And also I've mentored several MS bioengineering students, and they graduated from me. Some graduated first and worked with me. So just here a little acknowledgement. All the work would not have been possible without the students. So it is celebrating their contributions rather than mine, actually. And lastly, well, the title is The Past, Present, and Future of APTAMER. Where's the future? Well, present is the future. Now, we've just been through a tough time in the past 3 years since 2020 spring. And with a new disease, and it comes new advancements in technology. We talked about CRISPR a couple weeks ago. And here is a little update of how I, pull out a lot of the articles and put together and try to review how nucleic acid aptamers can be integrated with CRISPR-Cas technology. And several of those were related to detecting the problematic virus that is still lingering around. So that's why present is the future, to set the stage for the future. And really, lastly, something trivial. 2023 is the 33rd anniversary of phage display library, nucleic acid aptamer, SELEX process, and the commercializations of surface plasmon resonance instrument. Well, that's it. That's the end. Now, certainly there are a lot of biotechnology areas that I have not yet covered in this series, but I think this is a nice point for us to take a break. And if you like most content that you've been learning from me and want to hear more of my talk in the future, This Dr. Han's Pharmacy Classroom Pro will always be here, and I will periodically update older series and produce new series related to pharmacology, physiology, microbiology, and other broader biomedical science content. So maybe I will see you again in the future. And take good care. Bye.